Okay, well, good morning, or good afternoon, everyone. Just a reminder, I guess, that we are on the last day of our Term Quiz 1 rebuttal. So if you're planning to do a rebuttal, it has to come in before the end of the day. And then I'll try to endeavor to get those evaluated sometime early next week. And that's about it, I think. Are there any questions about things before we continue on with our lecture? Everybody's all good? Okay. Okay, so we made it about halfway through this slide yesterday, talking about the different organs that are innervated by the parasympathetic and sympathetic divisions of our autonomic nervous system, trying to get some sense of why the sympathetic and parasympathetic nervous system would have the various effects they do on these organs. So we covered the eye, the salivary glands, the lungs, the heart. Did we do the adrenal gland? We did the adrenal gland too. Okay, good. So the next organ on the list here to talk about is the liver. Somewhere, there it is, the liver. And the liver, as you can see here, again, is one of these organs that does not actually get a dual innervation. So the adrenal gland was the same way. We said it doesn't get innervated by the parasympathetic nervous system, quite possibly because epinephrine, once released from the adrenal gland, gets metabolized very rapidly in the body, has a very short half-life, don't really need to shut the adrenal gland synthesis down per se. Because of that, the liver is kind of the same way. It does not have a dual innervation. It receives innervation only from the sympathetic division, as you can see here. And it says that what the sympathetic division does to the liver is it stimulates glucose release. So the liver is a major storage site in the body for glycogen. And so when we are in this sympathetic mode, when we are very active, we need to mobilize the energy that is stored in the various parts of our body to make it available to the parts of the body, like the muscle especially, that are going to be very active during these periods of activity. So that's why the sympathetic nervous system is signaling to the liver to start taking the glycogen it has stored, hydrolyzing it down into individual glucose molecules, and then releasing those into circulation. We want to boost the blood glucose levels when we're active in order to power, to have a fuel source readily available to power activity. So that's why the sympathetic division is stimulating the liver to break down its stored glycogen. Why doesn't it have a dual innervation? Why doesn't the parasympathetic nervous system perhaps innervate the liver and have it build glycogen back up, for example, from the glucose that's in the blood? I imagine the answer to that question is it's a bit of a defense against hypoglycemia, right? Because ultimately, even if you're in a resting condition, you yes, aren't using up maybe as much glucose now, but also we don't want to just automatically signal to the liver to start taking up glucose from the blood and turning it into stored glycogen, because if there isn't incoming glucose from the diet, then that's going to ultimately lead to hypoglycemia. It's going to lead ultimately to tissues like the brain being starved of the nutrient that they require. So the liver basically takes up glucose and builds it into glycogen in a concentration-dependent manner, right? Only when the blood glucose kind of gets above normal levels and when it's not being stimulated to break it down, the glycogen it contains, when you're kind of in a resting state, but you're also having this high blood glucose level, that's the condition under which the liver will start to build glycogen and store it. We don't want it to just be automatic that it's trying to build glycogen from blood glucose every time we're resting, because again, if there's no influx of glucose to offset that glycogen synthesis, you could become hypoglycemic. Continuing on here, so we move on to the various digestive organs. So the stomach, the pancreas, and the intestines here, all involved, in course, in digestion. And so here it mentions that the sympathetic nervous system is going to inhibit the digestive organs, whereas the parasympathetic division is going to activate those digestive organs. Again, that makes sense, considering that when we're active, when we're in sympathetic mode, we want to be shutting off processes that don't contribute directly to our activity, like digestion. And when we're in parasympathetic mode, right, we're resting. That's the time we do these basic housekeeping functions like digestion, and so we want to turn those systems on at that time. Next, it comes up to the urinary bladder here, and it says that the parasympathetic division stimulates urination, whereas the sympathetic division inhibits urination, right? So generally speaking, when we are resting, we want to basically promote good blood flow to the kidney, right? Try to filter our blood, remove any waste, perform water balance activities, all these things. And so at the same time, it's going to promote that urine production, but also promote the release of urination as well. So your parasympathetic division promotes urine formation and urine elimination, whereas the sympathetic division does the opposite, right? When we are active, again, we want to be directing our blood flow to our muscles, to our brain, to our heart to power that activity. We don't want to be directing a ton of blood flow into our kidneys, so we don't want to produce a lot of urine. We certainly don't want to be releasing urine while we're active. Hence the sympathetic division inhibits that urination process. There is one kind of asterisk you could add here, is that right when you, if you imagine, for example, that you had your sympathetic division being turned on kind of out of a fear response, right? Kind of what's the word I want here? Like the, why can't I think of what I want to say here? In any case, if you have like a fear response, right, that's kind of like a startle, and that's what activates your sympathetic nervous system, oftentimes when you get startled, you do urinate quite a bit, right, at that point, right? Somebody who's like, peed their pants when they got scared by something, right? And you might say, well, okay, if you scared me and my sympathetic division turns on to kind of, mobilize my body systems to help me, fight or flight was what I was trying to think of earlier, right? You're in this fight or flight mode, you've been startled, your sympathetic nervous system is turned on, you're trying to mobilize your body systems, you're trying to get ready to possibly fight or run away from whatever threat you have. Why do we urinate right at that point when we're told that the sympathetic division inhibits urination? It's because when you have that kind of emotional, fear-based response, and that's what's kind of kickstarting the sympathetic division, the limbic system actually overrides the sympathetic inhibition on urination and promotes urine release prior to the fight or flight. And there are a whole lot of reasons why you could do this, not just human-specific reasons, just kind of broadly animal-specific reasons. But one is that if you have to run away, well, it'd be nice not to be carrying a giant bag of urine inside of you. It's a little bit lighter if you piss it all away and then run off. Some animals will actually use the urination that they release when they're afraid as kind of a chemical spray, or just to kind of create odors that might deter predators and things. So yeah, we've kind of got this evolved characteristic where if what turns on our sympathetic nervous system is kind of an emotional response, a fear response, being startled by something, our limbic system actually causes us to urinate right away. Whereas if you just get up and run out the door, you're probably not going to pee as you get up to run out the door. Just the sympathetic nervous system turning on inhibits urination, as we've said, but if it's turned on by more of a fear or startle response, it gets overridden and you'll actually urinate immediately. And then lastly here, it talks about the genitalia. And so the parasympathetic division, it says, promotes the erection of the genitals. So that would involve both the penis and the clitoris. And then the sympathetic division is promoting ejaculation and vaginal contractions. And here I think the simplest way to kind of make sense of these inputs is to just think about, any sexual act you've ever either participated in or watched or read about, or however you've engaged with sexual material and things in your life. When you think about getting an erection, right, typically you're kind of in this restful state at that point, right? So you're just looking at something, or you're with someone, you're kind of resting, you're getting aroused, you get an erection. It makes sense that your parasympathetic division, which is kind of on while you're in this restful mode, would be promoting that erection. And then if you think about the times when you would start ejaculating or having these vaginal contractions or an orgasm or something, hopefully by that point, you've been actually kind of working away for a while, and so your sympathetic division is highly active. So I think it makes sense then that you go from a parasympathetic system causing the erection to the sympathetic system causing the actual ejaculation, vaginal contractions, orgasm, just because that reflects that transition from kind of starting that sexual act in a kind of restful condition and ending it really, hopefully, in much more vigorous ways. But also, interestingly, of course, knowledge of these relative innervations can also help with treatments for things like premature ejaculation or erectile dysfunction, right? Because sometimes those conditions are caused by problems with the sympathetic or parasympathetic innervation of the genitalia. Okay. So we move on from that figure to this figure here. So one of the things I wanted to just draw your attention to, and so I kind of slipped this figure in here, it seemed like a good place to put it. You know, we've been talking about, you know, all these different parts of the peripheral nervous system, right? So somatic sensory neurons and visceral sensory neurons. We talked about them yesterday. You know, the visceral motor neurons that are part of the autonomic nervous system, the somatic motor neurons that are innervating our skeletal muscles. We've talked about all these different nerve fibers. And what I thought I would throw in here at this point is you're looking here basically at kind of the spinal cord and in this case, a spinal nerve that is emerging from that spinal cord. And if you look at the spinal nerve, so the definition of a nerve is that you have a bundle of axons within the peripheral nervous system. Okay, let's start there, just to make sure we have a definition of this term. A nerve is a bundle of axons within the peripheral nervous system. So here, we can see we have a spinal nerve, and we can see the different colored axons that are within this nerve. Again, some are somatic sensory neurons and visceral sensory neurons and so forth. Because we have a collection of both sensory and motor axons here within these spinal nerves are referred to as mixed nerves. They have both motor and sensory fibers within them. So all of your spinal nerves are mixed nerves. They have both sensory and motor axons within them. The cranial nerves, which we're not going to get into any detail about here, but if you've studied cranial anatomy, cranial nervous anatomy before, you'll know that the cranial nerves, some of them are mixed, but some of them are purely motor and some of them are purely sensory, all right, just as an aside. But in any case, you can see the spinal nerve here, and again, it's a mixed nerve, but what they're also highlighting here is as the spinal nerve is approaching the spinal cord, you'll notice how it splits into a dorsal root and a ventral root, and then kind of connects into the spinal cord. And what you can notice here is that the dorsal root, and therefore the dorsal horn here of the spinal cord, this is where all the sensory axons go, right? So the dorsal root and the dorsal region of the spinal cord are kind of dedicated to sensory information, whereas you can see here that the ventral root and therefore the ventral horn of the spinal cord is actually dedicated to motor neurons only. And so that's one thing worth kind of noting, right, is this kind of division of the sensory and motor information flow, shall we say, as we are approaching the spinal cord, going from spinal nerve to the roots and then the spinal cord itself. And so, yeah, I just wanted to kind of drop that information in there somewhere. Okay, so where I want to go next here, and where we'll spend the rest of our afternoon discussing, we've talked now about every part of this textbook figure on the nervous system and its organization, but there's one additional component to the nervous system that wasn't here in this figure, and it's very common that this part of the nervous system is ignored in a lot of human physiology textbooks, and that is what's called the enteric division of the nervous system. So as it says here, the enteric division is a network of nerves in the gut wall, and it promotes things like peristalsis, segmentation, and secretion within the gut. So peristalsis, you probably know, is kind of like a wave of muscle contraction. In the case of the GI tract, you have a wave of muscle contraction that'll pass from kind of your mouth down through to your anus to help kind of push food through the length of the digestive tract. Segmentation is more of this like back and forth sloshing kind of muscular contraction where you're trying to just slosh food back and forth within the gut. That helps to sort of mix in digestive enzymes, for example, helps to just maybe physically digest food. And then secretion, as we'll see in a moment, involves things like, acid secretion into the stomach, digestive enzyme secretion, even hormone secretion. There are a number of hormones that the gut secretes that some of which, communicate how much, food is in your stomach and things like this. Now, the thing we have to appreciate about the enteric division of the nervous system is it is capable of acting entirely independently of the entire rest of the nervous system, right? And so we've known for a long time, as the next slide will indicate, that, when you eat, and so there's like food present in your gut, right? There are sensory receptors in the gut that can detect the physical and chemical presence of the food you've eaten, and then that stimuli directly causes the muscle action and the secretory action of the gut. There's no requirement for that stimuli, that sensory information about, hey, there's food here. There's no requirement for that information to be sent back to the central nervous system, right? So when you think about most of the sensory structures in your body, and based on what we talked about yesterday, most often your sensory structures are picking up this sensory information, they're sending it back to the central nervous system for integration, and then the central nervous system sends out motor outputs to the various parts of the body to respond. But that's not happening here with the gut. That's not happening here with the enteric nervous system. The stimuli that the gut is detecting without any involvement from the central nervous system is generating a motor output response within the gut itself. All right? So that's important point number one. What you'll notice here, however, is I do have these sort of dashed lines drawn in, kind of showing us that there are inputs going into the enteric nervous system from these other parts of the nervous system. So although the enteric nervous system is capable of acting completely independently, it still makes sense for it to have some inputs coming to it to allow its function to be regulated in conjunction with what else is going on in the body, right? But what I want to emphasize is that if you were to come along and cut all those connections away, the enteric nervous system would continue to happily function on its own, okay? Now, in terms of the inputs that are coming into the enteric division from other parts, we can see that the sensory division of the peripheral nervous system, you can see, long way around, it sends signals to the enteric division. And that makes sense because ultimately, let's say you are sitting here and you start to smell food, right? Or you see food because somebody, well, actually no one seems to be eating, right? I don't know, maybe you've all eaten already, but sometimes you'll see food, you'll smell food, and you want to have your enteric division turn on even in advance of the actual consumption of that food to kind of like get your gut prepared for the food that you are going to probably eat, right? So if you smell food, you see food, you may not necessarily be able to eat it. You might be looking at her with whatever she's got and thinking like, oh, I can't eat that. I can't go over there and sip on her straw. But ultimately, that sensory input could still lead to the activation of the enteric division and therefore the activation of the gut in anticipation that perhaps I'll go get my own drink after lecture is over. So we have this sensory input that can regulate the enteric division, and then we also have both sympathetic and parasympathetic inputs, right? And that makes sense too because we want to be able to control the activity of the digestive tract in a way that fits with what else is going on in the body. Right, if we're currently in sympathetic mode, as we've already discussed, we want to be able to slow down the digestive function, whereas if we're in parasympathetic mode, we want to be able to speed it up. So we do have these inputs going into the enteric division to regulate it, but again, this system is capable of working entirely on its own. And this is not something we discovered yesterday. As my comment here says, we've known since the mid-1700s, right? That the intestine, for example, there were studies done back at that time which showed that you could take segments of the intestine from a vertebrate and it would respond directly to stimuli applied to it, right? Even if you had taken it completely out and it was acting completely disconnected from the brain or the spinal cord. And so we've known for a very long time that the intestine and the gut is able to respond directly to stimuli without the need for the central nervous system to be there. And so what people often describe the enteric nervous system as is they describe it as a second brain, right? It's kind of its own independent nervous entity, if you will, able to detect sensory information, integrate it together right on site, and then execute motor output. So that's why some people refer to the enteric nervous system as the second brain. Just to kind of anatomically introduce you to this enteric nervous system, on the left here we have a fairly generic illustration of the GI tract. So here you can see right in the very center is the GI lumen, right? So that's the open space through which food and water is passing. That is lined by a region of the gut called the mucosa, right? So that's what's lining the lumen. And that, of course, is the layer that is then actually absorbing nutrients into the body. Underneath the mucosa, you have what's called a submucosa. They don't per se label it, but it's sort of this sort of layer here sitting underneath of the mucosa. And then you'll notice that kind of just underneath that is where you have what's called the submucosal plexus or Meissner's plexus. So this is this yellow network of neurons that's underneath of the submucosal layer here. This is part of the enteric nervous system. Then underneath that, you have two layers of muscle, right? Circular and longitudinal muscle. So this is what does the peristalsis and the segmentation we were talking about. And in between those two muscle layers, you have another nerve network called the myenteric plexus or Auerbach's plexus. So that's the nature of your enteric nervous system, these two nerve networks, the submucosal and myenteric plexus, or Meissner's and Auerbach's plexus. And as they do try to illustrate here in the figure, they are connected, so they're not separate networks. They are two interconnected networks, but this is the nature of your body's enteric nervous system. And the figure on the right just basically shows it again from a different perspective. What I'm going to show you is this figure here, because this figure again tries to show the different parts of the enteric nervous system and again tries to show how this thing can function again completely independently of any other nervous system input. So we're going to start here. So again, this is the mucosa. Right, so this is the layer that is facing the gut lumen, where all your food and water that you've consumed is. And what you'll notice is in the mucosa, you have these mechano and chemoreceptors. And so the function of those receptors is to detect the presence of food within your gut. So the mechanoreceptors are detecting the presence of food just based on its physical presence, right? That there's like some physical entity in there. And then the chemoreceptors are detecting the presence of food based on its chemical structure, right? They can detect that there are fats, they detect glucose, they detect amino acids. So we have these two types of receptors to detect the presence of food. Now, of course, they can be fooled, right? I mean, your mechanoreceptors, if you were to just sit here and eat pieces of Lego, right, you're going to stimulate those mechanoreceptors. They're just going to know there's something physical here in the gut. They don't know that it's Lego that you stupidly ate and it's not food. And your chemoreceptors, again, they're a little bit more specific generally because they're targeted to detect specific kinds of nutrients generally. But anyways, you're trying to detect the presence of food based on its physical and chemical properties. And then once you detect the presence of food, then you'll notice here that we can trace pathways from these mechanoreceptors directly to the muscle layers, right? Again, without any involvement of the central nervous system, right? So as soon as we detect the presence of food in the gut, the neurons that are within that nerve network, the submucosal and myenteric networks here, those mechanoreceptors are again directly attached to the muscle layers to allow for peristalsis and segmentation to occur in response to the food that is present here. And there's no requirement that we send that information to the central nervous system and then have it send motor output back. You'll also notice here that these mechano and chemoreceptors, in addition to being connected to the two muscle layers, they're also connected to endocrine cells. And there are endocrine cells in your gut, right? So endocrine cells are hormone-secreting cells. There are a number of different hormones that your gut produces, right? Things like ghrelin, for example, which your stomach will pump out when you have no food. So in the absence of these mechano and chemoreceptors being stimulated, in that case you'll get ghrelin being released. And so when the mechano and chemoreceptors detect food, they'll shut down the secretion of those endocrine cells. But there are other hormones too. I don't want to get into them all here, but there's a ton of hormones that your gut produces, right? Either producing them when you've eaten certain nutrients or producing them when your stomach's empty. But the whole point of these hormones is to communicate to the rest of the body about the current food and nutrient load in your gut, right? So the body can kind of regulate what it's doing based on, have I eaten recently? Is there food in there? What kinds of nutrients are in there? What's going to be absorbing into the body soon? What do I have to deal with? All these hormones coming from the gut allow for that communication between the gut and the rest of the body, so the rest of the body can know what's going on in the gut and can act appropriately. And then you can also see how these mechano and chemoreceptors are directly connected to the various secretory cells. Like I said, there's acid-secreting cells in the stomach. We have cells that are producing various digestive enzymes and have to secrete them. So yeah, those are the major components there of the enteric nervous system. But again, I'll just point out here, they again highlight here in this figure the sensory and parasympathetic and sympathetic inputs that are coming here to the enteric division, right? Again, allowing for this independent nervous system to be regulated by what else is happening in the body, right? So in some ways you can kind of think of your cells maybe as like an enteric nervous system, right? You're independent people who do your own thing in response to your own stimuli, but maybe mom and dad have a say sometimes, Like that's the way you can kind of think of it. They're the central nervous system. You're thinking, I'm going to drop out of UTSC. Nobody's teaching me anything worth knowing. And they're like, no, I don't think that's a wise idea. Maybe stay in there, right? So there's independence, but regulated independence, one way to think about it. And so, again, I always say you can kind of come here with a knife if you wanted to and kind of slice through here, right, and cut off all those connections between the enteric nervous system and the rest, and it will continue to function just fine in the exact same way that if you finally do cut the umbilical cord that connects you to mom and dad, you will be just fine, right, without their influence per se. Anyways, I'm not trying to make you feel bad about your parents. All right, I want to go into a little bit more detail here on the enteric nervous system because, again, a little bit more that you know about the enteric nervous system, the more you can start to understand some of the pharmacology that's associated. Pertoy's not here. He was always hounding me about why aren't we teaching more pharmacology, and then when I bring it up, he's not even here to hear about it. He'll see it in the video. He'll reach out. In any case, what we see here is kind of similar to what we were seeing before. So again, you have your mucosa here, right? And again, they're highlighting here receptors. They mention these receptors can detect, the distension of the gut. So that's kind of a mechanical or physical expanding of the gut because of just the physical presence of food. And we've already said there are chemoreceptors that can detect specific nutrients. These sensory receptors that are in the mucosa, they are ultimately connected to this particular type of neuron here that they call an IPAN. And you can see here that stands for the intrinsic primary afferent neuron. So these are basically the sensory neurons, if you will, of your enteric nervous system, these IPAN neurons. They are the ones that are connected to the various mechano and chemoreceptors. Now what you'll notice that in turn, these IPAN neurons, they have axons that connect them to two other kinds of neurons, two kinds of motor neurons. Here in green we have the ascending excitatory motor neurons, and then down here on the other side in gray we have the descending inhibitory motor neurons. So the way this works, so let's imagine that there is food right here in this location within our gut lumen, right? So these mechano and chemosensors are detecting that, and they're sending a signal to this IPAN here within our myenteric plexus. What this IPAN then does is it sends signals down to these excitatory motor neurons. You can see here that they are located orally relative to the food. So further back towards your mouth compared to where the food is. And they're excitatory. So that means that they are going to in turn stimulate the muscle layers to contract. And then the same IPAN neuron detecting food here sends a signal down to these descending inhibitory motor neurons. They're more anally directed compared to the food itself, and they're going to inhibit these muscle layers and cause them to relax. So what are we accomplishing by doing this? Well, let's imagine, right, that we've got a piece of food in the middle of our gut there, and obviously we want to be pushing that food towards the anus, right? We don't want food being brought back up to our oral cavity unless we have to vomit for some reason, then we have to do things differently. But if we're not vomiting, we're just kind of eating food and having it pass through to our anus at some point, the way we accomplish that is when the food is in a given location, we contract the muscles that are upstream, right, back on the oral direction, we contract those, and then we relax the muscles that are downstream towards the anus. And so by contracting upstream and relaxing downstream, you are effectively pushing the food in that anal direction. Right? You squeeze, it's like if you had a straw or something, right? If you had some piece of something stuck in the middle of a straw, you squeeze behind it, right? Not in front of it to kind of push it out. That's what we're accomplishing here. So whenever we detect the presence of food in a given location in the gut, we are going to have the oral upstream muscles contract, the anal downstream muscles relax, so we can push the food in this oral to anal direction, all right? Now, what I also want to talk about here, I suppose, a couple of things on this slide. You can also see here, here's an IPAN, again connected to sensory receptors, in this case also connected to secretory neurons, as you can see. So those are the other major neurons as we discussed, right? The secretory neurons for secreting acids and digestive enzymes. And here they give us a list of the three major neurotransmitters that these secretory neurons use to communicate with the secretory cells that they are stimulating, right? So acetylcholine, which we have some familiarity with, nitric oxide, NO, which we are definitely familiar with for its immune context anyways, and then VIP, which it says is the vasoactive intestinal peptide, right? So those are the neurotransmitters that are being used by secretory neurons in your gut to communicate with secretory cells. But I also want to focus here, they tell us the neurotransmitters that are used by these excitatory motor neurons and these inhibitory motor neurons. You can see the excitatory motor neurons use acetylcholine and also substance P, and then the inhibitory motor neurons use a whole collection here, nitric oxide, VIP, neuropeptide Y, and also ATP, right? I don't know if you've encountered this before, but we always, of course, think about ATP mostly in its energy currency context. We've already learned about it as a DAMP earlier in the course, and now we're also learning that it's a neurotransmitter in some circumstances. So ATP's got more roles to play than often people are aware. Now, again, what's the purpose of talking about any of this? Like, why even bother to learn about any of these neurotransmitters that are involved? Well, because, as I kind of alluded to earlier, there can be some pharmacological relevance to this. Let me introduce you, if you're not familiar, to a condition called achalasia. This is a condition where what often happens is these inhibitory motor neurons within your digestive tract, they get lost, they die off. And so all you have left are the excitatory motor neurons that are causing muscle contraction to occur. And so if that happens in some region of the gut where you have just contraction and no relaxation mechanism, you end up with achalasia. So what you're looking at here is actually an X-ray. So they will have had this person swallow this kind of contrast fluid. So it would show up on the X-ray. And you can see, this is the esophagus here. You can see how the fluid is building up here because you have this region right here that's extremely contracted. The muscle there in the gut is contracted and it won't relax and it won't really let much fluid pass. I mean, here you can see a little bit of fluid is passing, but not much. It's so contracted. So obviously this is not good. If you have esophageal achalasia, it's like you drink some juice and it literally just gets stuck in the esophagus and then is, in this case, trickling out maybe over time, right? Not an ideal circumstance. So how could we try and treat this? Well, let's exploit what we learned from just a moment ago. One of the most common treatments for achalasia is this compound here called isosorbide dinitrate. Because when a person consumes isosorbide dinitrate, which I believe you can take orally, don't quote me on that, but I'm pretty sure that's the route of administration, you can see when you look at its structure that there are these nitric oxides that you could imagine being liberated through metabolism from that compound. So when you take isosorbide dinitrate, it basically boosts the nitric oxide levels within the body. And then, of course, as we saw, nitric oxide is a neurotransmitter promoting muscle relaxation in the gut. So now we can hopefully get some relaxation of this region that is overly contracted to allow for foods and liquids to pass through the digestive tract. So there's an example of where we can kind of utilize the information that we know about the enteric nervous system in some kind of clinical way. Okay, where I want to go next here, kind of keeping the enteric nervous system in mind, is I want to start thinking about what's called the gut-brain axis, which is this idea that there is this kind of special, intimate relationship between our gut and our brain. And let me start by kind of showing you, again, kind of the pathological, I guess, implications of this relationship. This is a study, data set here from a study where researchers looked at individuals who had either no depression, right, or they've been diagnosed with mild, moderate, or severe depression. And when they measured the incidence rates in these individuals of these two different digestive disorders, so functional dyspepsia, which is basically kind of like a chronic upset stomach, and inflammatory bowel syndrome, you can see that anyone who had depression had much higher incidence rates of these two digestive disorders compared to those who were not clinically depressed. And so, yes, obviously this is just a correlative data set, but it does tell us that there is some kind of relationship between dysfunction in the brain and dysfunction in the gut. They seem to have some kind of relationship. And again, I want to emphasize that this is not something that we just learned yesterday. This is something, in fact, if you go back, Hippocrates, at least it's been attributed to him. We don't have to debate if that's true or not. But this notion that all disease begins in the gut, right? 340 BC. Like, we've known for a very, very long time that there is this significance to gut problems manifesting in problems other places in the body, and in particular in the brain. It seems like at some point we kind of forgot about this gut-brain connection, and only in the last decade or so have we kind of rekindled our interest. And one example I can use to show that is this paper here. You can see 2019 in this journal called Psychiatric Times. They had this review paper talking about nutritional psychiatry, the gut-brain connection. And the idea here is that if you look at the field of psychiatry, right, from, say, the 1970s through probably to the 1990s for sure, maybe later, the psychiatry was very focused on pharmacological solutions to psychiatric disorders, right? It was always, you have this kind of psychiatric disorder, we'll prescribe you this pharmacological agent, and it will correct your problem. And where the field of psychiatry seems to be shifting a lot more now is to this idea that nutrition can be a powerful way to regulate or to treat a lot of these same psychological disorders. We are starting to recognize more and more that some of the psychological and psychiatric disorders that we suffer from are the result of our poor diets, really. And so, many people do espouse the belief that it's the shitty Western diet of heavily processed garbage that we eat that might partly be underlying all of the mental health disorders that we see cropping up at higher and higher incidence rates. So, if you take nothing else from, this Friday afternoon with us here, one might be eat better, feel better, right? Which, of course, you didn't need someone to teach you necessarily, but maybe just someone to remind you, right? That if you are feeling depressed, if you are feeling like, I've got some of these mental health disorders, think a little bit more about, well, what am I eating, right? And could I be eating in a way that's better? We're seeing lots of evidence to show that link. What I want to talk about next here is I want to expand out this gut-brain axis just a little bit to include the gut microbiota. So now we have this microbiota gut axis, and so this is this idea that not only is there an intimate relationship between our gut and our brain, but this is the idea that that relationship is mediated in large part by the commensal microbes that inhabit our gut. Right? So we got introduced to those commensal microbes back in our lecture on immunity. We're going to be reacquainted with them here and now start thinking about how the kinds of microbes I have in my gut could be influencing this gut-brain axis relationship. And so I'm going to use this figure here to guide this conversation. And where I'm going to start here is kind of right in the center of this figure, where it talks about how, if these commensal microbes we have in our gut, if they find their way into our body somehow and our immune cells detect them, then our immune cells, as we learned before, will respond by pumping out cytokines, and these cytokines can actually lead to alteration within the brain that could alter things like mood and cognition and emotion. So let's explore this a little bit here. The first thing to think about is, well, why would these microbes suddenly be coming into the body, right? So we know again that we have to have these commensal microbes in our gut, right? We already talked about the important roles they play in immunity. They have important roles to play in digestion, like they're necessary things. The problem is when they get inside the body, when they leave the gut lumen and they cross the epithelial barrier and they come into the body. Now, normally, they're not able to do so because as the top photo or top figure here shows, here's your intestinal epithelium. So there's all your commensal microbes here, and then here's the body underneath. Normally, we have this very tight barrier, this epithelial, intestinal epithelium. This normally serves as a very tight, leak-proof barrier. And in fact, you can see here how they're talking about tight junctions that are expressed, right, to basically bring the plasma membrane of neighboring epithelial cells here in the intestine so close together that nothing can pass through. These microbes can't get through the intestinal barrier. Nutrients and things can, because we have transporters to facilitate their passage, but microbes are not able to come into the body from the intestine into the body because there's no passageway for them to go. There's no transporters to invite them in. There's no space between the neighboring epithelial cells of the intestine because of the tight junctions. They are kept in the gut lumen. But then what they show us in the bottom figure is that if that intestinal barrier becomes leaky, if you lose those tight junctions, or if the cells of the intestinal epithelium begin to die, right? Here's an enterocyte undergoing apoptosis. If you lose those cells or you lose those tight junctions, you end up with a condition known as the leaky gut. And suddenly these commensal microbes that were restricted to your gut lumen are coming into the body, and then they're interacting with the immune cells, and the immune cells produce cytokines, and those cytokines impact the brain function. Well, obviously the thing to think about here then is, well, how would we go from having nice intact intestinal epithelium to the leaky gut? What's going to cause that to happen? And so what we have to appreciate is how much energy it takes to maintain the integrity of our gut lining. I don't know if anyone ever took B32 with me, but we spent some time in there talking about how, guts are some of the most expensive tissues to maintain. In fact, it's so expensive that in some snakes that only eat like once a month, they build their guts when they need them for that one meal a month, and then they tear them apart and get rid of them the rest of the time because they're too expensive to maintain. Well, here it says 40% of our body's energy, and this would be resting values here, 40% of our resting metabolic rate is directed just to the maintenance of our gut, to the maintenance of this intact barrier. And so what can cause the leaky gut? Well, basically anything that redirects energy away from the gut, because then the gut doesn't have the energy supply it needs in order to maintain its integrity, and its chances of becoming leakier go up. And you'd be surprised how long the fucking list is here of things that can actually cause the leaky gut that can do this. And again, most of them are because of how blood flow is being redirected away from the gut. So here they list, for example, chronic stress, whether physiological or psychological. Chronic stress is a known cause of leaky gut because when we are stressed, we activate our sympathetic nervous system, which, as we saw at the beginning of class today, directs the shutting down of the gut, right? And so we reduce the blood flow to the gut. We're not bringing enough energy there for it to maintain its integrity. So chronic stress, right? Strenuous exercise. Not that I've been trying to discourage you from ever doing exercise. The last 2 days I think I've been doing that. But strenuous exercise, again, because you're directing blood flow to muscles and to heart. You're taking blood flow away from the gut. Hyperthermia, right? There again, you're decreasing blood flow to the gut because you're trying to direct a lot of blood flow to the surface of the body to dissipate excess heat. Pathogen infection is not about blood flow. Pathogen infection is about bacterial toxins and how they can directly damage the intestinal lining. High-fat diets, and this is because when you eat a lot of fat, you have to then secrete a lot of bile salts to help with that fat digestion. Well, bile salts are helpful in the sense that they facilitate fat digestion, but they're actually also themselves pretty toxic. And so they actually can cause damage to the gut lining as well, right? There's a strong connection, for example, between also like colon cancer and high-fat diets, which we think bile salts are also carcinogenic. They mediate that relationship. NSAIDs, we've already talked about them. We know how they're going to cause damage because they inhibit prostaglandin production in the gut. They inhibit that protective mucus barrier. Alcohol consumption is on the list here. I don't exactly know the mechanism. And then pregnancy, for again, the fact that you are directing blood flow to the developing fetus rather than to your own gut. So again, there are a lot of factors here that can cause this leaky gut, that can cause the commensal microbes in our gut lumen to enter our bodies and come into contact with the immune cells. And then, as I said, once you have those microbes coming in and interacting with the immune cells in response, as we learned, they start pumping out cytokines. And so let me end here with this thought about cytokines. I'll just cover it kind of briefly, and then we'll have to pick it up on Tuesday. But what they're showing us here in this figure is how peripherally released inflammatory cytokines, so cytokines coming from immune cells outside the nervous system, like we saw here, and I'm hoping that if you look at this list that they've given here, TNF-alpha, IL-1, IL-6, that you'll be like, yeah, I've seen this list before. These are the M1, right, M1 cytokines that are pro-inflammatory. It mentions here how these pro-inflammatory cytokines coming from other parts of the body, they enter into our brain and then they cause brain cells like microglia and astrocytes to start making their own pro-inflammatory cytokines. And so the leaky gut basically leads to neurological inflammation. Now again, I'll just briefly mention this point and then I'll shut up and we'll pick it up on Tuesday. But it mentions here in this little figure that there are both positive and negative aspects to neuroinflammation. But what I really want to just highlight for you right here as the parting thought is if you look at negative aspects of neuroinflammation, notice what you see, the words high and the words chronic. Chronic high neuroinflammation, which could occur because you've got this leaky gut and this constant interaction of gut microbes with your immune cells, that's going to be negative. That's going to lead to things like anxiety and depression, to cognitive impairment, to neuronal damage. So if we have this leaky gut and we have this chronic commensal microbe going into the body, prompting immune cells to make cytokines chronically and at high levels, that's going to be bad. Right? A little neuroinflammation is actually a great thing, as we'll talk about Tuesday, but a lot of it is not good. All right, we will stop there. We will pick this up on Tuesday. So enjoy your weekend, and I'll see you next week.